Hello, my name is Gleb Bezgin. I'm a research associate in the labs of Professors Yasser Medina and Sharif Karama at McGill. And today I will talk about longitudinal cortical thickness agent trajectories and their association with cognition and lifelong experiences. I have no disclosures. The main motivation for this study is that healthy aging is a universal incentive. I'm humbled to give this talk on the birthday of my father, who is turning 70, and despite all my efforts to pull his hair off, he's still going strong. Another motivation is that cortical thickness is a robust predictor of biological age, either linear or nonlinear depending on age range. This relationship is mediated by multiple factors, including genetics and cognitive performance. Longitudinal data and analysis cannot be replaced by their cross-sectional counterparts, but they can be informed by them. And multivariate approaches may help tease out patterns from such multidimensional data as those that come from neuroimaging. The main aim of the study is to identify factors shaping heterogeneous aging trajectories. For that, we use two cohorts. The longitudinal Lothian birth cohort 1936, and for training the model, the cross-sectional NKI-RS lifespan cohort. The methods involve multivariate inference of aging trajectories, and we will describe the results in the context of longitudinal subtypes and their association with subject phenotypics. All subjects in Lothian birth cohort 1936 were born in 1936, administered a battery of tests at the age of 11 in 1947, and had many years later multiple waves of scanning starting age 70. We use data from the fully completed 4 waves. The NKI-RS data are representing most of the lifespan cross-sectionally, while LBC 1936 is a longitudinal data set starting around age 71 until age 82 in our study. The MRI preprocessing followed standard steps, namely T1 data were subjected to free surfer, thickness estimates were mapped to TKT atlas, and the resulting data were harmonized using COMBAT before subjecting it to longitudinal trajectories analysis. The analysis method is known as CTI, Contrastive Trajectories Inference, developed in our lab. It takes multidimensional data, MRI in our case, as an input, reduces dimensionality by applying contrastive PCA. For that, it uses so-called background and target. Usually, in the context of background and target, in the context of the CTI analysis, we talk about pathology-free versus advanced pathology stages. But because in our data, we use only cognitively unimpaired subjects, the background is represented by young adults with the age range of 30 to 40. And the target represents the oldest subset of subjects older than 77 years old. An important piece of information that we can extract from CTI analysis is pseudotimes, a model-based age estimate, normalized between 0 and 1. The first analysis is done actually with the pseudotimes and thickness, and specifically difference between pseudotimes and difference between thickness values across longitudinal waves of scanning. We observed inverse correlation between longitudinal change in pseudotimes and longitudinal change in cortical thickness for all regions. This result can be interpreted the greater the model-based aging estimation is, the greater the cortical thinning. From model training using cross-sectional data, CTI analysis gives us heterogeneity tree with each subject assigned to one of two trajectories. For now, let's call these trajectories blue and red. In the longitudinal context, a subject may appear assigned to blue trajectory across all waves or to the red trajectory across all waves. It may also happen that the subject would monotonically change from one to the other, for example, from blue to red or from red to blue. And this change can be non-monotonic. Let's call this a mixed subtype. Showing this with actual data, these subtypes can be referred to as generally higher thickness and generally lower thickness, consistent across all waves, and subtypes in which a subject monotonically changes across waves from high to low, fast decreasers, and from low to high, slow decreasers. A mixed subtype represents something in between these four and is not shown here for simplicity. Superimposing this data on the brain surface, we see that the difference between low and high cortical thickness subtypes is quite evident. For fast decreasers, the rate of decrease every 3 years is more than 2%, whereas the rate of decrease every 3 years for slow decreasers is less than 1%. So the difference is also evident. We observed relationships between subtypes and phenotypic data. In this plot, the higher values indicate slower decrease in general intelligence, the G factor. This specific G factor was calculated using fluid intelligence values for all the subjects across the age of 70 to 82. And the higher values here indicate slower decrease in general intelligence, and lower values indicate faster decrease in general intelligence. We can see that it's generally slower decrease in general intelligence for higher thickness and slow decrease, slow cortical thickness decrease subtypes, and generally faster decrease in general intelligence for low thickness and fast decrease subtypes. Further, ApoE4 allele frequency is higher for low thickness subtype than for all the other subtypes taken together. And the number of cigarettes smoked was higher for low thickness subtype than for high thickness subtype. To conclude, thickness trajectories investigated using LBC 1936 and NKIRS MRI data in this study. CTI analysis identified two agent trajectories with lower and higher thickness. And these two cross-sectional trajectories informed 5 longitudinal subtypes: generally high, generally lower, fast decreasers, slow decreasers, and mixed. Estimated subtypes had different phenotypic characteristics, as observed here via change in intelligence scores, ApoE4 status, and smoking data. As a general conclusion, cross-sectional data are informative for longitudinal model estimates. And concerning future plans, we plan to include data from new waves of scanning, including omics data. Thank you very much.